Fish sizes and habitat variables, exploring the connection between environment factors and fish length by Jordan Moonen, Emma Zelenak, and Laura McKay. Fish are a diverse group as they inhabit various water bodies, including oceans, seas, rivers, lakes, and ponds. Fish length among various settings is an interesting problem to study because fish species exhibit a wide range of adaptations and strategies in response to environmental conditions, such as temperature and water quality. Factors such as temperature and water quality can vary immensely, and fish can be present in these varying conditions. Therefore, studying what environments fish populate and the weather conditions will provide us with an understanding of why their lengths vary, as well as the type of fish that populate that area. Studying variations in fish length allows us to appreciate the diversity of fish species and their abilities to thrive in different habitats. Fish contribute to nutrient cycling, control population sizes of other organisms, and serve as a link in food webs. We hypothesize that how do environmental characteristics relate to fish length?
So the population we are attempting to simulate consists of all fish within our data. From the EPA data set, we selected a sample of 50 individual fish representing 10 different regions. We chose to research 10 different regions because researching several regions helps avoid biases that may arise from studying only one specific location or population. Researching various regions can also help us identify patterns, trends, and disparities in fish populations. By comparing these areas, we can gain valuable insights into the factors influencing variations in fish length. Within the EPA data set, we decided to include dairy livestock, PFAS, temperature, and site type, such as urban or non-urban sites, due to the varying impacts they can have on fish length. For example, pollution in agriculture runoff, which can be due to site type or can result from dairy livestock, can be specific environmental stressors that can affect fish growth. Temperature is also an appropriate variable to measure since it can alter the metabolic rates, which can change the growth patterns of fish. We decided not to include the variables of latitude and longitude because they don't always apply broadly across different locations. In contrast, studying common issues like temperature and pollution can provide insights relevant to many water bodies worldwide. We used a t-test to compare differences in fish length based on whether the sites were urban or non-urban. We used an ANOVA to compare differences in mean temperatures based on the region. And we used a histogram to compare differences in fish length frequencies. We used a frequency distribution to compare differences in cow dairy livestock across different locations. And we used a critical alpha value of 0.05 to compare our p-value to determine significance within our data.
In figure 1, the mean fish length can be represented as 362.039 plus or minus 14.362, and the range can be represented as 486. In table 2, the mean livestock can be represented as 5,047 plus or minus 1,139, and the range can be represented as 28,424. In figure 2, the mean temperature within regions was 13.2392 plus or minus 0.70948, and the F value can be represented as 33.741 with a p-value of less than 0.001, showing a significance between temperature and regions. Figure 3, the mean fish length can be represented as 362.039 plus or minus 14.362, and the t-cal can be represented as negative 0.288 with a p-value of 0.387, showing no significance between site type and fish length.
In conclusion, our sample size we used was 50 fish across 10 different state regions. We conducted an ANOVA on the mean temperature across state regions and found that there was significance with a p-value of p is less than 0.001. That is then compared to the critical alpha of 0.05. We also found that the F value was 33.741, which is much higher than 1, also indicating significance. After completing the t-test, we did not find any significant findings as the p-value of 0.387 is greater than our critical alpha value of 0.05, indicating no statistical significance. Our t-test was fish length present at different site types, and findings showed that there was no difference between the fish length and their site type. We failed to accept our hypothesis based on the analysis conducted.